Welcome to Anatomy and Physiology. This is lecture number 2 in Bio 177. So we're going to talk about vertebrae, talk a little bit about chemistry here. So let's begin by talking about the body plan of vertebrate animals. So first of all, a vertebrate animal is an animal that has a spinal cord. So that's basically a type of nerve fiber, nerve structure, along with the actual bone, which we consider the stiffened supporting structure next to that spinal cord. So a couple of different animals, humans, reptiles, amphibians, birds, fish, they're all considered vertebrae, and they're also going to share another common feature. Okay, they're all going to have one or more cavities within their bodies. So we're going to take a quick look at the early development to see what happens here. So in this first image here, if you're looking at the zygote, and then it kind of shows the development of the zygote. So again, when you get an egg cell from the female and the sperm cell from the male, the sperm cell fertilizes the egg. You get what we call a zygote. Okay, a zygote is that fertilized egg, and this zygote is going to undergo repeated divisions, as you can see. So, it kind of jumped one of those stages on our image here. It jumped from what we can kind of see as that one cell stage to the 8 cell stage. But again, that's just the idea of what happens embryologically. And eventually we get so many divisions that we form what is a hollow sphere that you can see there. And, it's called a blastula there, as you can see. So the blastula is going to be that hollow ball made up of cells. And, the next picture there, you can see that the blastula has been cut open to reveal something called a blastocele. Okay, and essentially that blastocele is that cavity that has formed within the center of the blastula, right? So eventually that structure is going to fold inwards to form a gastrula, and that's what you're seeing down there on the image towards the left there, where the arrow kind of continues on. So that's the inwards folding that occurs to kind of change the blastula into a gastrula. So we call that gastrulation. And as you can see on the next image there, we're showing you the different parts that eventually form from the gastrula. Okay, so we can see 3 distinct layers. And as we continue on through our lecture series, we're going to sometimes, when we want to learn about the embryology of the subject, we're going to go back and talk about these three layers time and time again. So it's important to get a basic understanding of these now and get used to these terms. So the 3 distinct layers of that gastrula, therefore, are going to be the ectoderm, as you can see, and the ectoderm is going to be the outer layer. Okay, and the ectoderm is going to give rise to the outer covering of our bodies, like our skin, eventually, but it develops from the ectoderm. And then sometimes we get some parts of the nervous system as well. And then we also get the endoderm. Okay, and the endoderm is going to be the most inner layer. Okay, and that's going to basically going to be the layer that eventually is going to line hollow organs. It's going to line the hollow digestive tract, that endoderm. And then lastly, you get the mesoderm, and the mesoderm is the middle layer between the 2 other layers, and that's the layer that's going to end up forming most internal organs. Okay, so that's what you have there, the 3 different layers of the gastrula, the 3 distinct layers. And you can see that already one of those cavities is kind of forming within it, right? Okay, so let's go on here. All right, and then on this image here, it continues on talking about these body cavities that we'll be discussing soon, these coelums, and that alimentary canal that was formed just now by that gastrulation. And again, that blastula folds inwards, called gastrulation, creates fluid-filled cavities called coelums, and also creates the alimentary canal known as the digestive tube. You can kind of see the formation of what's happening there with that gastrula. So there are two important facts I want you to take away from this. There's going to be a hollow tube called the alimentary canal, which passes through the center of the body, leading from the mouth to the anus. Okay, now this tube that we're talking about, this alimentary canal, is not considered a cavity. It's actually considered a space that's considered outside of the body. So again, it's not really considered a cavity, it's a tube, and the space within it, the lumen, as we call it, the middle of that tube, is actually considered part of the outside of the body. Okay, and then there are several other tubes that are continuous with the outside. Okay, so again, if you think of the like how we mentioned, the mouth, the esophagus, the stomach, small intestine, large intestine, all those pathways, even though they're found on the inside of our body, they're considered continuous with the outside world. Okay, so hopefully that's clear. So they're not considered cavities. They're not considered closed off cavities. So during development of a vertebrae, such as a human, several large cavities, in fact, do develop. Okay. So again, here is an image of the alimentary canal from the mouth, stomach, small intestines, all the way to the anus. So again, this is part of it's considered, part of the outside world still. We call this the alimentary canal. And then remember that the inside of any tube is called the lumen. So the lumen of the alimentary canal would be considered part of the external environment. So let's actually talk about what a cavity is then. What are these cavities? What are these coelums? So during development, these vertebrae, such as humans, we're going to develop several large cavities that are filled with specialized structures. And there's two main cavities that we need to learn here. Okay, we're going to learn about the dorsal cavity, and we're going to also learn about the ventral cavity. Okay, so again, let's kind of talk about these cavities here. Now, before we actually go on to talk about the cavities themselves, there's something that we should discuss, and that topic is the serous membranes as well. Okay, so one thing about these cavities that we're going to be talking about, whether it's the dorsal body cavity or the ventral body cavity, these cavities are going to be covered by a thin double-walled membrane called serous membranes. Okay, so again, if we go back to this idea here, the dorsal body cavity that we're talking about, that's going to contain the brain and the spinal cord. Okay, so it's going to contain the cranial and spinal cavity, respectively. And then the other cavity that we were just talking about, the ventral body cavity, that's going to contain what we call the thoracic cavity and the abdominal pelvic cavity. And like I just mentioned, except for the cranial cavity, which is going to be a little bit specialized, we'll talk about that later, these cavities are considered coelms. And like we mentioned earlier, they're derived from mesoderm. And some of these structures, some of these cavities are going to have specialized membranous structures that we're talking about that are called serous membranes. And these serous membranes are going to enclose that cavity, and within these serous membranes, we're going to find a fluid filled space. Now, the serous membrane that encloses the organ and connects it to the body cavity wall, that's basically its function. And each serous membrane is going to have an inside and an outside layer with specialized serous fluid that we're going to find in between these two layers. So again, if you look at the first image here, you're looking at an example of what one of these serous membrane cavities might be like. And the analogy that it's giving you here is you've kind of blown up a balloon, halfway here, very lightly, and you've stuffed your fist into the balloon. Okay, and you can imagine that the hand in this case is the heart, it's going to be the organ, and the balloon that you find around the heart is going to be one of those serous membranes. And that structure, the hand along with the balloon structure, is going to fit, let's say, inside the chest cavity. Okay, that it fits inside the chest cavity. So you end up getting the heart, then you end up getting a layer of membrane that's touching the heart, then you get a hollow space that's filled with air in between the two, and then you get another layer of membrane, which in this case would be touching the inside wall of the chest cavity. Okay, now in this case, this actual structure has a name. This exists if we were talking about the serous membrane that covers the heart, and that's going to be called the pericardium. Okay, so the pericardium is the lining that we find in the pericardial cavity that lines the heart. Okay, so again, the two layers that we need to learn about here and memorize of these serous membranes, we're going to have a parietal serous membrane layer, and the key word there is parietal. The parietal layer, that's the one that's going to line the inside wall that we talked about. So with the pericardium, it was the layer that lined the inside of the thoracic cavity. And then the other layer that you need to learn about is called the visceral layer. Okay, the visceral serous membrane. And in this case, that was the layer that was covering the heart, that was covering the organ, that was covering the viscera itself. So very important to remember those two distinct layers, the parietal and the visceral layer. And remember that the different cavities in the body are going to be covered, the organs are usually going to be covered in these serous membranes. And in our analogy, when we were talking about the fist entering the balloon, that air that we talked about earlier, that would actually be filled with fluid inside the body that we would call serous fluid, that would help to support the organ, help to cushion it. Okay, so in the image there below, you can see a little bit more of a more realistic representation of the heart and the pericardium and the relationship that the heart has to those two layers. And then finally, the last picture in the set there, it's a really zoomed in view where you can see the different actual layers of the heart. Finally, going down to the very, very like most intimate layer that it's pointing to, which is known as the serous membrane. So that would be the layer that we would probably consider that the visceral layer of the serous membrane. And then you have that space in between the two where it's pointing to the watery fluid, which reduces friction. And then on the opposite side of that would be the parietal layer. Okay, so on this one here, like we mentioned, if you're looking at that little red structure on the top left image, that's supposed to be a deflated lung. And you'll notice that the black structure, which is the cavity, is lined with that blue line. So the idea behind this is you can imagine that you're stuffing the deflated lung into the cavity, and you know, as you stick it into that cavity, you're pushing it in, and a part of that membrane kind of intimately wraps around the organ, as you can see. And then that black area would be the empty space that would soon be filled with fluid. And then on number 3, you see that we've inflated the lung, and you can see the two intimate layers of that serous membrane and that very thin black layer in between the two now. That would be that serous layer that's filled with the serous fluid. So we're going to call that the pleura. So the pleura is the serous membrane of the lung. And the pericardium we talked about, that was the serous membrane of the heart. So a couple of images there describing these structures. The visceral pleura, that would be the pleura in contact with the lungs. The parietal pleura, that would be the layer in contact with the chest cavity. And then in between the two, we would have the fluid, right? Okay, so we've given a couple of examples here. Let's move on to the next cavity here that we're going to call the peritoneum. Okay, so again, the peritoneum, if we're looking at this image here, you can see that the abdominal cavity has many organs, and once again, these organs are going to be covered by a visceral peritoneum. We're going to get a space filled with fluid in between the two, and then we're going to get a parietal peritoneum. Okay, and then another important thing to mention here is that there are some organs, such as the kidneys, that are actually not contained within this peritoneum. They would be considered retroperitoneal, meaning that they're behind the peritoneum. So to kind of summarize what we've talked about here, okay, we've got 3 serous cavities that are enclosed by serous membranes. These 3 serous cavities are going to be the pleural serous cavities that we mentioned that contain the lungs, the pericardial serous cavity that contains the heart, and then the peritoneal serous cavity that contains the digestive organs, some accessory organs of digestion such as the liver, the ovaries, the uterus, and the superior portion of the urinary bladder. And then we talked about retroperitoneal being behind the peritoneum, between the peritoneum and the muscular wall of the abdominal cavity. And we said that a couple of organs that we might find retroperitoneal were like the kidneys or the pancreas. Okay, now, we're going to go ahead and leave behind the gross anatomy of the human body, and we're going to focus more of our attention on the chemistry of cells for this next portion of the lecture here. Okay, so we'll start by talking about elements and atoms. So in order to understand physiology, we really need to know some biochemistry. Okay, so that's the chemistry of organic molecules as they are related to different biological processes. So to do this, to understand more about chemistry and therefore the cells and physiology and such, we need to review some basic concepts of atoms and molecules. Now, the first term I want to talk about is the term element. Okay, so an element would be considered the chemical building block of matter. Okay, so for example, different elements that we might find are those that we see on the periodic table. Okay, so a couple of different elements might be that of sodium, which we would abbreviate that as Na. We'll talk about that a little bit later. Okay, another element could be that of potassium. Okay, we'd abbreviate that as simply the letter K. There's hydrogen, okay, etc. So each one of these elements are going to exist as a unique type of atom, and atoms generally are going to consist of different parts. So they're going to consist of protons, which carry a positive charge, neutrons, which carry no charge, they're neutral, and then electrons, which carry a negative charge. Now, the way that these different elements are going to be represented on that periodic table will be how we can see on our image there below. Okay, so um, they'll be represented by what we call an atomic number. Okay, and that atomic number um, is the number of protons or electrons. Okay, and these are always equal, they never change. And they'll also be represented by an atomic mass. Okay, and that's the mass of the protons and the neutrons in the atomic, in the measurement of atomic mass units. So again, if we look at the image there above, if we bring our attention to the hydrogen, hydrogen is being represented as an atom that has one proton in the center, and then on the outside shell, you can see that it has one electron. Okay, if we go down to carbon, you can see that carbon has 6 protons, 6 neutrons, 6 electrons. Okay, nitrogen is going to have 7 protons, 7 neutrons, 7 electrons. Oxygen is going to have 8 protons, 8 neutrons, 8 electrons. Okay, so a couple of examples of different molecules there. And again, the important thing here, if we go down below, we can see that these molecules, these not molecules, these chemicals are going to be represented by, or rather these atoms are going to be represented by what we call a symbol or a name. In this case, hydrogen is represented by the H. We're going to have an atomic number. The atomic number is going to be the number of protons or electrons. And remember, those are always going to be equal. Okay, the number of protons and electrons are always equal. And then the atomic mass, which is the mass of the protons and the neutrons. So again, we're not going to be going so far out into chemistry, since the course is not really a specified chemistry course, but we do need to touch upon it here. So again, all atoms for an element are going to have the exact same atomic number, but they may not have the same mass. Okay, now sometimes an atom occurs in the form with a different number of neutrons, for example, and that's where we talk about these isotopes a bit. Okay, so again, sometimes an atom occurs in forms that are going to have different numbers of neutrons, and in these cases, these are going to be called isotopes. So, for example, the normal carbon would be considered carbon-12, but a commonly used isotope of carbon, and the reason why I say it's commonly used is because it's used to radioactively date fossils. Okay, so an alternative form of carbon would be called carbon-14, so that would be considered an isotope. So carbon-14 still has 6 protons, but it has 8 neutrons. So as you can see, that it doesn't follow that rule that we talked about earlier, where they should actually be equal. So carbon-14 would be considered unstable. It would be considered radioactive. And when it begins to break down, the word that we give for that is when it begins to decay, it throws off high energy particles in a process called radiation. Now, although all these radioactive isotopes can cause cell damage, many of these are actually going to be used medically to treat diseases and also for diagnosis. All right, so let's talk a little bit about molecular interactions and chemical bonds next. So atoms are often going to interact with one another to form molecules, and such interactions often result from atoms sharing electrons or other electromagnetic interactions that will form bonds. So let's talk a little bit about the different types of bonds that may form. Okay, now one of these types of bonds that may form is called a covalent bond. And a covalent bond would be considered a strong type of bond, and that's when electrons are shared between two atoms. So the making and the breaking of a covalent bond would require energy, and sometimes energy can be also released as well. So for example, when we hydrolyze ATP into ADP, we're going to release energy, and that energy is going to be used in different reactions in the human body. Okay, so again, covalent bonds are strong bonds where the electrons are shared between two atoms. That's the key word there, where they're shared between two atoms. Okay, so we have two basic types of covalent bonds. There's nonpolar covalent bonds and there's polar covalent bonds. So in nonpolar covalent bonds, the electrons are shared equally between the two partner atoms. In polar covalent bonds, we're going to have two poles that are formed. Okay, now sometimes one of the atoms in a molecule pulls a little bit stronger on the electrons in its bonded partner than its bonded partner. Now this is going to cause one part of the molecule to be a little bit more negative and the other part to be a little bit more positive. So as I mentioned, now we've created a type of polarity. Okay, now that's called a polar bond. And a great example of a polar bond would be like H2O or water. Okay, so again, the main idea here to understand here, please, is that covalent bonds are bonds that share electrons, and we have two types of covalent bonds. We have polar and nonpolar. Now, a little bit um, that I want to talk about as far as these bonds are concerned. Now, before I go on, I'd like to just mention about non-covalent bonds. So again, those are the bonds where we don't share electrons. Um, so in non-covalent bonds, those are bonds that do not involve the sharing of electrons, but rather involve different electromagnetic interactions. So a couple of different types of non-covalent bonds could be considered, such as an ionic bond, a hydrogen bond, and a hydrophobic um, interaction. So let's talk about these, and then we'll lead up to hydrophobic versus hydrophilic. So again, an ionic bond. Okay, in this type of bond, one molecular partner is going to pull on an electron completely away from the other. So we don't have sharing anymore. So that stronger partner then becomes completely negatively charged, whereas the weaker partner becomes positively charged. Now, each partner is going to be called an ion. And positive ions, we're going to call those cations, and negative ions, we're going to call anions. So an example of one of these types of chemicals would be, or molecules rather, would be sodium chloride. So that we would find, for example, salt. Now, in water, sodium chloride would dissociate into ions of sodium and chloride. And in this case, the sodium would be positive, the chloride would be negative. The sodium would be the cation, the chloride would be the anion. Okay? Now, another type of bond are hydrogen bonds. So this type of bond is going to involve a weak interaction between a partially positive hydrogen atom and an electronegative oxygen. Um, it can also be nitrogen or sulfur atom. Now, no electrons are shared. The bonds are weak individually, but they have a huge number there are a huge number of organisms that use these bonds that are important for life. So, for example, H2O attracts nearby water molecules, which would result in surface tension of water. and that's due to the hydrogen bonds that are formed in between those water molecules. Okay, now that brings us to this next concept here, hydrophobic interactions. So nonpolar molecules are going to tend to aggregate together in aqueous solutions to form to separate from water, which would limit the exposure of hydrophobic surfaces to polar water molecules. This leads to two important terms describing molecules based on this type of interaction. Okay, so we have hydrophilic versus hydrophobic. So a hydrophilic would be a water loving. That's what that term refers to. Water loving. Water loving. Hydrophobic would refer to water hating. So a hydrophobic interaction would be one that occurs between molecules that cannot both participate in hydrogen bonding, while a hydrophilic interaction occurs between molecules that can participate in hydrogen bonding. When we talk about, again, hydrophilic, we usually talk about proteins, which are hydrophilic. So again, hydrophobic, we said that those don't dissolve within water, and that could describe things like, for example, I'm sorry, not proteins, but lipids, including steroid hormones. And then the hydrophilic would contain things like would include things like proteins. All right, and then this thing here, it's just it's an app that's available. Essential Anatomy 5, have no affiliation with it, but it's a good app that might help in studying the human body. So you can download that if you'd like. Okay, and what we've talked about so far is we've talked about serous membranes. Main things that we talked about with the serous membranes is that they're going to fill. They're going to wrap around different organs. So we said that when we're talking about the dorsal cavity, which contains the brain and the spinal cord. We talked about the ventral cavity which is divided into thoracic, abdominal, and then the pelvic cavity. We talked about serous membranes, the parietal layer, the visceral layer, a little bit about atoms and isotopes, types of bonds, and hydrophilic versus hydrophobic. All right, let's move on here and talk about organic compounds. Okay, so despite more than having 110 known elements, our body is going to be comprised mostly of just 6 main elements. We're going to have carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Okay, and we can abbreviate those as, again, carbon as C, hydrogen as H, oxygen as O, nitrogen as N, phosphorus as P, sulfur as S, as in the periodic table. Now, most of these biological molecules, however, are just going to be made up of actually just 3 elements, which are carbon, hydrogen, and oxygen. So most of what is made of organic matter are just those 3 elements. Now, together, these 3 elements are going to be largely responsible for the thousands of organic compounds found in our body. And there are 4 classes of organic compounds in the body. There are carbohydrates, lipids, proteins, and nucleic acids. 3 of these, which are proteins, carbohydrates, and nucleic acids, are considered macromolecules. Okay, so those are considered macromolecules. And basically, those macromolecules can also be considered polymers, which or are made up of polymers rather, which are the single building blocks. Or rather, let me go back there. Sorry. So these macromolecules that we just mentioned are polymers, and what that basically means is that they're larger molecules that are made up of smaller ones, and those smaller molecules are called monomers. So let's take a look at what we're talking about in a little bit more detail here. So carbohydrates, for example, which is the first one that we're showing here. So carbohydrates, this is the most abundant biological molecule, mostly comprised of carbon, hydrogen, and oxygen. And it's going to serve as an energy source. Also, it's going to form certain structures. It plays a part in the building blocks of certain things, and even more. Uh, Now, larger carbohydrates are called polysaccharides and are comprised of a smaller building block called monosaccharides. So going back to that idea, the polymer in this case would be that large polysaccharide, and then the monomer would be the monosaccharide. Okay, so that larger carbohydrate, that polysaccharide, um, would be the polymer. The monosaccharides would be the monomer. And then some examples of monosaccharides could include things like glucose, fructose, ribose. Okay, and then glucose um, plus fructose equals sucrose. That's table sugar. Okay, that's basically what the body and cells are going to use as an immediate form of energy, mostly. And then glycogen is another term that we can talk about. Glycogen is formed by many glucose molecules binded together, and glycogen is how the body is going to store glucose. Okay, there's also lipids. Lipids are going to be a diverse group of molecules. So these are fats, essentially, and then these fats can be considered either hydrophobic or amphiphilic, okay, which means that they are both hydrophobic and hydrophilic. Now, unlike other organic compounds, lipids do not form large macromolecules, nor are they built for monomers. Instead, lipids are basically long chains of carbon and hydrogen. We call these hydrocarbon chains. And sometimes they're going to be linked with smaller molecules like glycerol. Now, we have three different types of fats, three different types of lipids, I should say. There are fats, phospholipids, and steroids. Now, um, as far as steroids are concerned, those are typically going to be chemicals based on fats that play a role in messaging systems in the body. In hormones is what we're talking about there. And then phospholipids, those are going to be a very important class of lipids that we'll talk about later that are going to form the basic structure of the plasma membrane, which is the covering of every single cell in the body. Okay, so again, fats. Fats are going to be a very efficient form of long-term energy storage. These are also functional in some places. For example, fat pads are responsible for the breast shapes, and we can find fat pads that serve as padding between organs. And fat can also serve as insulation. For example, there's a flap of fat that's found internally in the abdominal cavity known as the greater omentum. And fats are a large lipid comprised of a fatty acid, hydrocarbon chains linked to a glycerol molecule. And again, an example of that would be a triglyceride. And then the example of phospholipids that we talked about, that was a class of lipid that's largely responsible for the membranes that are around cells. Okay, we said that phospholipids are very similar to fats, but these are going to have two fatty acid chains that are linked to a glycerol backbone, which also contains a phosphate group, okay, made of phosphorus and oxygen. And we're going to talk about the importance of this molecule when we talk about cell membranes. Okay, and then we talked about steroids already. We said that this was a class of lipids where the carbon skeleton forms four rings instead of the elongated chains. The most basic form of a steroid is cholesterol, which is also found in many animal cell membranes. Now, many animals also are going to use it to make a class of important molecules called steroid hormones, and a couple of examples include testosterone, estrogen, cortisol, etc. Okay, now the next group of organic compounds are proteins. Okay, now proteins are polymers of amino acids, building blocks. So these are going to serve a tremendous variety of functions, from structural to communication to enzymes and more. Now, the structure of a protein is key to understanding its function. We can talk about the primary structure of a protein, a secondary structure, the tertiary structure, and the quaternary structure. And then I'll show you an image soon that I'll use to describe each one of those. And then the last type of organic compound here are nucleic acids. Again, these are comprised of carbon, hydrogen, oxygen, nitrogen, and nucleic acids are comprised of monomers called nucleotides. Individual nucleotides function in energy storage and transfer, whereas nucleotide polymers function in information storage and retrieval, such as in DNA. Monomers, okay, we said that these monomers, like what we're seeing here, are the individual pieces that come together to build larger pieces. Okay, so that process we call polymerization. Okay, so we get these smaller parts come together to form a larger thing, and then you see that larger thing there to the right side. That's that macromolecule. Okay, and then we talked about carbohydrates. Okay, carbohydrates were an excellent example of polymers and monomers. We said that the smaller building blocks, such as the monosaccharides, come together to form polysaccharides. And some examples of carbohydrates include glucose. That's probably the most common type of energy source for the human cell that gets processed in an organelle called the mitochondria, eventually to form ATP, which is the active form of energy in the human body. And we said that the storable form is called glycogen, which is stored in muscles and fat, which can be converted back into glucose when the body demands it. Okay, we talked about lipids, the triglyceride being a storable form of energy. Eventually, these triglycerides can be converted back into sugars. Steroids, which we said play a role in messaging in the human body, and then those phospholipids that you see to the left, which play a role in the membrane of all cells. Okay, we talked about proteins. We said proteins are made up of smaller blocks that are known as amino acids. So amino acids essentially come together to form larger structures called proteins. And we talked about the 4 different levels of structures of proteins: the primary, secondary, tertiary, and quaternary structure. So you can see that the primary structure, so the protein is going to be made up of a unique sequence of amino acid monomers that will then make up the polypeptide chain of the protein. Okay, now next what happens is that the protein needs to kind of get modified a little bit further. Okay, so we then form the secondary structure of protein. Okay, now the backbone of the amino acids can then create coils or folds that fold into polypeptide chains. Okay, that would then turn into the tertiary structure. Okay, now the functional group of amino acids can bind to one another, creating additional complex shapes on top of the helix or folds that were caused by the backbones. And then finally, the most complex structure of that protein that we have there is the quaternary structure. So finally, the protein could consist of 2 or more polypeptide chains, and these can be linked together to form different shapes as well. And the book goes to talk a little bit more about that there as well. Okay, and then finally, when we talked about nucleic acids, we said nucleic acids are going to play a role mostly in the formation of DNA and RNA. Okay, now the book also is important to read this chapter on. So remember to cover it on the book. I'm not going to go too much further into depth as far as this topic is concerned, but you know, do read the book, and then do send me any questions if you do have them. Thank you. That's the conclusion of this part of the presentation. We'll see you on the next one.